Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about the genome organization of E. coli K12. The genome of E. coli K12 is a single circular double-stranded DNA molecule containing just 4639 kilobase pair. The genome of E. coli K12 it contains 4405 genes, and the genome is localized in a specific area that is known as nucleoid. Comparing with the genome of eukaryotes, in E. coli there are more genes and less space, whereas in eukaryotic genome there are more space and less number of genes. In the genome of E. coli K12, introns are absent. What is intron? Introns are the non-coding sequences that are present in the genome of eukaryotes. E. coli has no discontinuous genes at all. Discontinuous gene means the gene which contains both coding sequence and non-coding sequence. However, the genome of E. coli K12 contains certain other sequences that might be repeated elsewhere. The sequences are insertion sequence 1 and insertion sequence 186. The circumference of the genome of E. coli is just 1.6 millimeter, whereas the size of the E. coli cell is very small. The size of E. coli cell is just 1.0 into 2.0 micrometer. So the genome of E. coli is packaged with the help of specific DNA binding protein so that the genome can be squeezed into a very tiny space inside the E. coli cell. Even in eukaryotic cell also, the genome being very large is packaged with the help of specific DNA binding protein so that it can be accommodated within the nucleus. In prokaryote also, the genome is packaged with the help of specific DNA binding protein so that the genome can be squeezed to fit into a tiny space inside the E. coli cell. The most important feature of E. coli genome is the E. coli genome is supercoiled. Supercoiling is of two types, positive supercoiling and negative supercoiling. When additional turns are introduced into the DNA double helix, that causes positive supercoiling. And when turns are removed from the DNA double helix, it creates negative supercoiling. In E. coli genome, the supercoiling is generated and controlled by two different enzymes. These enzymes are DNA gyrase and DNA topoisomerase 1. These two enzymes are found to be associated with the E. coli genome, where they generate and control the supercoiling. According to the current model, there is a central protein core. From the central protein core, 40 to 50 supercoiled loops radiate out. These are the supercoiled loops. About 40 to 50 supercoiled loops radiate out from this protein core. Each supercoiled loop contains about 100 kilobase pair of supercoiled DNA. These are the loops in which the DNA is supercoiled. 4 different proteins are found to be played a specific role in packaging of the E. coli genome. Out of 4 different proteins, the most abundant protein that play a specific role in packaging of E. coli genome is Who protein. Who is the most abundant protein present in the nucleoid of E. coli? About 60,000 Who proteins are present per E. coli cell. The Who protein is different from histone protein. Histone protein is a basic protein that play a key role in folding of the eukaryotic chromosome. Whereas Who protein, it play a similar role like that of histone protein. In prokaryotes, the Who proteins form a tetramer. Around the tetramer, 60 base pair of DNA is wound. So, with the help of various DNA binding protein, including Who protein, the DNA of E. coli or the genome of E. coli is folded in an organized manner so that it can form a very compact structure and it will be easily accommodated within the nucleoid. The nucleoid is the specific localized area in E. coli where the genome is found. Friends, this is about the genome organization of E. coli. For more updates, please subscribe Sam online class. Thank you very much friends. See you again. Bye bye.